So thanks for coming. So I have the pleasure of introducing Anna Nagel, who's been a student in my lab for the last 4.5 years. So Anna's finally making her exit. She's actually made several exits over the years since the pandemic started. And she's driven back and forth to Minnesota at least 8 times with her father, who's been very gracious and done the drive with her often. And this time, hopefully, this is her final exit from California. So Anna completed an honors degree in biology at the University of Minnesota. She minored in mathematics and chemistry. And while she was there, she also did an internship in computational biology at Los Alamos, working with Thomas Leitner and Nick Hengartner, who work on HIV phylogenetics. So that kind of part of the sort of genesis of her interest in HIV and phylogenetics, which we're talking about. She also did an undergraduate thesis project in theoretical population genetics with Emma Goldberg and Unique Brandvain. Some of you, if you've been in Eve for a while, you might remember Unique. He was a postdoc here years ago. She arrived as a PhD student in my group in the fall of 2019 on the brink of the COVID pandemic. She brought with her an NSF graduate research fellowship, and she also received the Dean's Distinguished Graduate Fellowship, all of which made it difficult for me to spend money on my NIH grant. Fortunately, she's GSR on my grant this quarter, so I'm actually going to spend down some of my huge budget. Anna chose to work on basically implementing phylogenetic methods for Bayesian tip dating. And if you haven't heard of Bayesian tip dating before, you're in for a treat because Anna's going to tell you all about it. Her motivation initially for working on that was that she was interested in HIV biology and applying phylogenetics to HIV sequences within patients. And so with that type of data, the samples are taken at different points in time, and the virus evolves very quickly. So you actually have to take account of when the viruses were sampled in order to do the phylogenetics correctly. So that sort of became the focus of her research. And part of it was sort of addressing this broader question of how you deal with tip date samples correctly in the context of these phylogenetic models. So basically in the course of that, Anna really got deep into sort of programming. She's actually the first student in the lab who's contributed code to the program BPP, which I co-developed with Song Yang and Thomas Flores. So Thomas is actually a programmer by profession. He is actually a systems programmer. So the program is written in a very low-level C language. And so Anna really developed a lot of low-level programming skills as part of her project. So she's sort of a programmer as well as a biologist now. Anna's work so far has led to one paper in the Journal of the Royal Society Interface, and that's on estimating ages of latent lineages in HIV patients, and she'll be talking about that. And she also has another paper that's in review currently in systematic biology, which deals with essentially phylogenetic methods for estimating trees using ancient DNA. And she'll be talking about that as well. So I want to say a couple of things about Anna. First of all, Anna loves Minnesota. You know Anna at all. She'd love to talk to you about Minnesota. And be careful because you may end up buying property on Lake Minnetonka. She's also a circus aerialist, okay? And the great thing about that is that without being rude, you can tell Anna to take a flying leap. She's also the first student in my lab to knit me a double helix toque, which is totally awesome. I just have to show you guys this toque. There we go. Okay, without further ado.
I didn't know my Inuit was going to get an introduction, but thanks for the introduction. And as Bruce mentioned, today I'm going to talk to you about the work I've been doing on Bayesian phylogenetic inference using data genetic samples. So I'm going to spend a good portion of this talk talking about methods we can use to estimate time-calibrated phylogenies. But I think a good place to start is to ask, why do we need time-calibrated phylogenies? It's a lot easier to estimate phylogenies that are not calibrated in some sort of units of time like years. And often the reason is because we need time-calibrated phylogenies to answer other questions we care about. And so this is an example of work by a fellow Pop Bio student, Jay, and they're interested in looking at COVID dispersal rates over time. And so this is data from the beginning of the pandemic in China. And on the x-axis here, we have time, days, and on the y-axis, we have a number of viral dispersal events. So Jay is trying to estimate number of viral dispersal events over time and in relation to these other events that we know by date, such as changes in policy. And so you can see that these changes in viral dispersal events seem to be correlated with or potentially driven by these changes. But we couldn't start to address this question unless we first had a phylogeny calibrated in units of time. We had it in some other units, we couldn't look at how the viral dispersal events changed over calendar time, which is what we understand. And so this is one example. Another example is if you want to understand something about diversification. And so if we look along the edge of the tree, these are a bunch of different mammal orders. And these authors were particularly interested in understanding whether these orders arose before or after the KPG boundary, which is basically the extinction of the dinosaurs. So the gray is the extinction of the dinosaurs. And so to ask this, we know when the extinction of the dinosaurs was in some units of millions of years. So we need to turn this tree into units of time. And again, on the inset, we have time on the x-axis and number of lineages on the y-axis. And we can look at the number of lineages of extinct and extant mammal genre over time. So lots of reasons. In this case, these authors are interested in understanding diversification over time. So these are just a few examples. You could come up with a lot more examples of why you might want to estimate time-calibrated phylogenies. But I'm going to talk about how do we actually estimate them. A lot of the ways we estimate time-calibrated phylogenies start with a molecular clock. So a molecular clock is the idea that the amount of molecular evolution is approximately proportional to time, with the caveat that if you have long periods of time, you need to account for multiple substitutions at a site. So if we look at this graph, on the x-axis, we have time in millions of years, and on the y-axis, we have amino acid replacements per site. And then we have a bunch of pairs of species, and we can see that this relationship between time and amount of replacements is approximately linear. So this means that the rate of evolution is approximately constant over time. But you might notice if I were to go out and collect sequence data, the only thing I could actually measure with sequence data alone is the amino acid replacements per site. If I just have sequence data, I can't tell you anything about time or rate. And so this information from time in this case is coming from fossils. So if I have molecular data, I can make a tree that only is in units of expected number of substitutions like this, or mutations. So we have time, the number of mutations, and something a molecular clock predicts is that the tips of this tree, if I have all extant organisms, are all going to line up at the bottom because they've all been evolving the same amount of time and the rate is constant. But let's just say for the sake of argument, I'm just going to pick a mutation rate, and then I can change this tree into units of years. But if I picked a different rate, I could shrink the tree or expand the tree proportionally, and all the trees are equally good if we don't have any information about rate. And so the story with tip dating is a bit different. So in this case, we'll think about something like a virus where we're going to sample one virus earlier in time and some viruses later in time. And so if I did the thing I just did where I'm going to change the rate and rescale the tree, you might notice that my date changed, but I just told you I know when I sampled that date. So I can't do this because I'm breaking the assumption that I know when this tip was sampled. And so this is telling us that when we have samples taken at different points in time, this gives us additional information about rate and time. And so for my presentation here, I'm going to be talking about two very different systems. I'm going to be talking about viruses, and in particular HIV, and mammoths and other types of ancient DNA. And these two are not united by some aspect of their biology, but rather how we can use the sample ages or sample dates in our analyses. So I'm going to be talking about sample ages or tip dates kind of interchangeably, and I'm going to mean different things when I talk about viruses and ancient DNA. So for viruses, I'm literally going to mean when did someone go to the doctor, get their blood drawn to get their HIV sequenced. So it's like the calendar date. When I'm talking about ancient DNA or mammoths, I'm meaning the time that the organism died. And this can be estimated through means such as radiocarbon dating, which uses the ratio of different isotopes of carbon to estimate how long the organism has been dead. So it's not when someone went out and sampled them, it's when the organism died. And so the idea of tip dating works as follows. So with viruses, we're going to sample a bunch of viruses, and then we're going to put the viruses on this time axis. So all the tips, they line up with when we sampled them. And then we can use the branch length in combination with these different tip ages to try to estimate these nodes' ages in the tree. So the node ages in the tree are on the same time axis. So with viruses, this is often the only option. We don't really have outside information on the ages of internal nodes in viral phylogenies. For something like a mammoth, we actually often do have outside information. We might have fossils. So in this case, these authors estimated a time-calibrated tree using molecular data, molecular sequence data in combination with these fossils to try to estimate this time-calibrated tree. But using fossils is inherently really challenging. And so tip dating could be seen either as an alternative to using fossils, or it could be used in combination with fossils to try to estimate a phylogeny informed by both. So now for my talk, I'm going to be going into my projects, and they both are Bayesian phylogenetic tip dating methods. And they both make use of a computational technique called MCMC to try to sample from posterior distributions that we can't solve for analytically. So this is all very technical, and I'm not going to describe it at all. You're welcome if you didn't want to hear about it, but if you did, you can talk to me later. So now I'm going to go into my first project, which is on HIV. We need to learn a little bit about HIV. So HIV is a retrovirus, which infects human immune cells. In particular, infects CD4 positive cells. So when someone is infected with HIV, the HIV will bind to the host cell. It will fuse to the host cell membrane. Its RNA genome will be reverse transcribed into DNA. The DNA is integrated into the host cell genome, and then it uses the host cell replication machinery to make copies of its genome to make proteins. These assemble and go butt off to go infect new cells. And so when a patient has HIV, they're typically treated with a combination of antiretroviral drugs. So these might target specific stages in the life cycle, such as fusion, reverse transcription, or integration. But some of these cells, these drugs can't target. And these are latently infected cells. So basically, these are long-lived cells that have HIV integrated into the host cell genome, but they just stop at this integration phase indefinitely. So they retain the ability to finish this life cycle, but they might just wait out if this integration phase for years or even decades. And so what this looks like within a patient is when a patient has HIV, they're going to have virus in their blood, which I'm showing with these little green things. They're going to have cells that are actively infected with HIV and making more virus. And then they're going to have some of these latently infected cells. And so if a person is treated with a combination of antiretroviral drugs, this will get rid of all of these viruses and all the cells that are actively infected, and they'll be left with these latently infected cells. And so, as I said, these cells retain the ability to complete their life cycle. So they could become actively infected. And if they're not taking their antiretroviral drugs, this will very predictably lead to a rapid rebound in viral load and disease progression. So due to the clinical relevance of this pool of latently infected cells, people are really interested in understanding various characteristics about it, such as when did individual HIV proviruses become latent? So this is a question I'm going to seek to address in this project. I'm going to address it through a phylogenetic standpoint. So for the sake of argument, let's just say I could go out and I could collect some HIV sequences from a patient, and I can collect some latent sequences. And I'm going to collect them today and make a tree in units of time, in years. So if I did this, the tree would look like this, and all the tips are going to line up because I collected them today for this little thought experiment. But if I instead made the tree in units of mutations, the tree would look different. This branch to the latent sequence is going to be much shorter. And this is because latent sequences have a much, much lower mutation rate than actively replicating sequences, because they basically just have their DNA sitting around in the host cell, not accumulating mutations at this fast rate that most of the lineages are. And so this difference in branch lengths is going to be the signal that I'll use to try to estimate these times. So my goals for this project are to develop a simulation method so I can simulate both active and latent HIV lineages, and then to develop a method to estimate latent integration times, and then lastly to compare with other methods. So I'm developing the simulation method explicitly so that I can test my method. I'm not developing it like as a project in its own right. This is to test the other methods. So we need to know a little bit more about HIV. So HIV has a really characteristic viral load over time. So we'll look at the x-axis, we have time going from weeks to years, and then we'll focus on the red line, which is on the HIV viral load or RNA. So this is just a measure of how much virus there is in the body. And we see that a few weeks after infection, there's this really high peak in viral load, and then it falls several orders of magnitude. This is a log scale, so this is a big change. And then there's this period of approximately constant amounts of virus before eventually the onset of age, which is shown at the end. And so because there's basically just so much HIV in the body during this peak period, people think it's really important in establishing this latent reservoir. So we'd like to have dynamics that mimic these dynamics in our simulation. So I'm developing a simulation method based on an existing body of literature of ordinary differential equations. I'm developing a stochastic simulation, and I'm not going to tell you any of the details, but I'll tell you what I'm tracking. So I'm going to be keeping track of 5 different types of cells or viruses. So first, we're going to track target cells. So these are CD4 positive cells that could potentially become infected with HIV. We also have virions, so this is basically virus in the body that can go infect cells. We have cells that are actively infected with HIV, so they're currently making more HIV. We also have latent replication competent cells. So these are cells that they have HIV integrated into the host cell genome. They're not currently making more HIV, but they retain the ability to do so. And then lastly, we have latent replication incompetent cells. So these are cells that have HIV integrated into the host cell genome, but they actually can't make fully functional HIV. And this is due to defects in their genome. And I'm including them for 2 reasons. One, we can't tell them apart from the replication competent ones unless we have whole genome sequences, which we usually don't for HIV. And also they're really abundant. There's a lot more of them than the replication competent ones. So if we're going to go sequence them, we're going to get a lot of these ones in our sequencing data. And so the different arrows here represent different possible transitions. So this infected cell can become, or this uninfected cell can become any of the 3 infected cell types. This latent cell can become activated and start making virus. And then all the out arrows show that these viruses and cells die at their own rates. So this method will give me times of events and counts, and I can use it to produce genealogies. So in this simulation, I will get a tree topology with branch lengths, and I'll know the amount of time each lineage was latent and the type of lineages that were sampled. So the way this works is we're simulating forward in time and we're starting at the root of the tree. So an actively infected cell can branch off to produce a virion, as shown here. Virions can die. Virions can produce new infections. These infected cells can also die. And they can also produce latent infections, which can potentially reactivate. So this is my simulation model. And if I parameterize this model with empirical values, I get dynamics that look something like this. And so if we'll just focus on this middle plot here, which is the virions. On the x-axis, we have time in days, and the y-axis, we have viral count on a log scale. And we see that we get this similar dynamics to what I showed before, where we have this peak phase and then approximately stationary amount of virus. So this suggests that I'm getting the dynamics I expected with this simulation. So this is great. I now have counts and trees, but I actually want sequence data to use in the method, not just tree topologies. And so I also wrote a DNA simulation program. And I'm using a traditional DNA substitution model, which is an HKY plus gamma model. And I'm making the assumption that no mutations can occur while a sequence is late. And so I'm simulating in the traditional fashion, which is to start at the root of the tree and simulate forward in time towards the tips. And so anywhere there are these dots, we just assume that no mutations can occur. So this sums up my simulation method. And again, I'm using this to test my other methods, which I'm going to talk about next. So next I developed an inference program to have a Bayesian phylogenetic model to estimate these latent integration times. And I'm starting with an existing program called MCMC tree, which can estimate time-calibrated phylogenies for viral sequences sampled over time. So to this program, I'm going to add a new parameter, which is the latency times. So in this little toy tree here, I'm going to have two sequences that are not latent. So they're actively replicating B and C, and then I'll have one latent sequence. And we can estimate this latency time as anywhere between the sample time and the age of the parent node, so anywhere in blue. And so for every tree, I'm going to tell the program which sequences are latent, and I can have as many as I want, and which are not, and then we'll estimate a latency time for each latent sequence. And we have to know beforehand which sequences are latent, which is empirically not a problem. So this is the basic phylogenetic model. And I've also developed a method to combine estimates across loci. So HIV has a single piece of DNA that will be integrated into the host cell, and it's very short. So there's one integration time for the whole genome because there's just one piece of DNA. But there's a lot of recombination within HIV, which means that different regions may have different topologies due to a history of recombination. So if I were to sequence, for example, this region called P17 and NAF, the trees might have different topologies. So we'd like to be able to share data across these different regions, but allow for the topologies to be different, which is not usually what viral phylogenetic programs do. So I developed a method to do this, and I'm not going to tell you the math on how we do it, but I'll present some results on what it looks like. So on the x-axis here, we have latent integration time in thousands of days, and then on the y-axis, we have the posterior probability density. And this straight line is showing the true value, because I'm going to be presenting simulated data, so we know the truth. So I'm showing here 4 different regions that I simulated. From top to bottom, they get smaller. So C1, V2 is the largest, and TAT is the shortest. And we see that for the shorter ones, we have a pretty flat distribution, and this is reflective of the prior. We just don't have very much information in the data from these sequences. But for the longer sequences, we have more information. We have a more peak distribution, and it's pretty close to centered at the true value. But if we now use this new method to combine across different regions, we get a more peak distribution that is again centered at the true value. So when we combine across these different regions, we get more information than using any one of the regions alone, even the longest one. So the next thing I wanted to do was to compare with other methods. So I'm comparing to 3 other methods. One is based on a least squares dating approach. One is based on linear regression, and one is based on pseudo-maximum likelihood. So now I'm going to show more simulated results. So, or simulation results. So I wanted to see how these results vary based on the alignment. So these sequences are short, we might get different inferences with different alignments. So I have one tree topology with 20 latent sequences, and then I'm going to simulate 30 different alignments. And so on the graph here, we're going to have the true integration time on the x-axis, and then the inferred integration time on the y-axis, and then we have the different methods across the top, with mine being the Bayesian method. So if these methods work perfectly, all the points are going to fall on these red lines. You can see they don't work perfectly. There's quite a bit of spread across the true line. In particular, we see that these 2 methods have quite a bit more spread, and this is because they don't have bounds. So this linear regression method, we can infer the sequences became latent after they were sampled, and potentially before the patient became infected, which is just not biologically realistic. In contrast, these 2 methods do allow bounds, so that's why they look more constrained. So the next thing I wanted to do is I wanted to look across different tree topologies. So I calculated a statistic, for example, the bias for all the points in this Bayesian plot. And then I repeated this 300 times. And that's what I'll be showing on the next plot. So we're looking at 300 different tree topologies, but we're averaging across one of these panels. So on the x-axis, we have the different gene regions. So C1v2 again is the longest, cat is the shortest. All is the method to combine across the regions with all of the 4 regions or just the 2 shortest. The different colors are the different methods, and then we're looking at the bias. And this dashed line here shows no bias. So what we can see is that when we have long sequences, so over here at C1v2, the methods are not that biased, none of them are. But as we get shorter sequences, they're more biased. Our method is more positively biased, the other ones are more negative. In our method, we know the bias is from the prior. We're less clear about the other ones. Next, we looked at root mean square error, which is a good way to compare across methods because it combines the variance and the bias. And what we can see is that our method consistently has lower root mean square error than the other methods, which suggests our method is performing favorably. Next, we looked at the size of the 95% credible intervals or confidence intervals. I'm not showing the maximum likelihood because it doesn't provide confidence intervals. And so basically if you have smaller confidence intervals, you get more precise estimates. And so what we can see is that the Bayesian method and the least squares method have similar size of the confidence intervals, but this linear regression method has much larger confidence intervals, potentially being on average like 15 years, which is really large for something like K2. Last, we looked at the probability the true time is in the 95% confidence interval. So if the methods work well, we expect this to be 95% of the time, which is shown with the dashed line. So what we can see is that the Bayesian method and the linear regression method seem to have pretty good coverage, which is close to 95%, but this least squares method has much lower coverage. And so combined, our method has smaller credible intervals, but we still have the true time in the 95% critical interval close to 95% of the time, which suggests our method is working well. So lastly, I wanted to analyze some empirical data. So on the x-axis here, we have the inferred integration time in years. I'm going to be showing 3 lines on each panel. The dashed line is the time the patient became infected. The solid line is when they began treatment, and then the dotted line is when they were sampled. And then these are different regions of the genome across the top, with this last one being the method to combine across different regions of the genome. And so what we can see here is that for the individual regions of the genome, we have pretty wide credible intervals, but we have much smaller credible intervals when we use this method to combine the estimates, which gives us a lot more information because we have much smaller credible intervals. We also see that the points appear to be clustered loosely around the time of treatment initiation, which has been an argument in the empirical world. So in conclusion, we have a method that has greater accuracy and precision than many of the alternatives. And we also have provided a way to combine inferences across different regions of the genome without assuming topologies are shared. And there are no other methods that provide this. So now I'm going to shift gears. I'm still talking about tip dating and Bayesian phylogenetics, but we're now going to talk about mammoths. So for a long time, people have been able to go out and find ancient specimens, such as this mammoth tusk shown here. But more recently, with advances in sequencing technology, we can actually get pretty good quality DNA from some of these ancient specimens. And as I referred to earlier, we can use this data potentially in combination with fossils to try to estimate time-calibrated phylogeny, such as this elephant mammoth phylogeny shown here. But if we think about these samples, these are inherently from organisms that lived potentially thousands of years ago. There's an age associated with them. And we know from a long history of work with viruses, one, that these ages can provide us additional information about rate and time, as I mentioned, but we also know that if we don't account for the ages of the sequences, we can potentially bias our divergence time estimates. And we also know from history of work on the multi-species coalescent that if we're thinking about closely related species, like mammoths and elephants, for example, we should be thinking about the relationships between gene trees and species trees. So this is pretty central to this project, so I'm going to walk through this. So I'm showing here a species tree, and this has 5 species or populations, also use species and population interchangeably. So we have population A, B, and C, and then we have the common ancestor of B and C, and the common ancestor of A, B, and C. So if I could go out and I want to sample one individual at one locus for each of the 3 extant populations, and I want to ask how are those samples related to each other. And so we know the samples can't be related to each other until they occur in the same population. So we start at the tips of the tree and we look backward in time until they're in the same population. In this case, we look backward in the time into population BC. And there's 2 possibilities. The sequences could share a common ancestor or coalesce in population BC, or they could not share a common ancestor, which is what happens here. And so we look farther back into time into population ABC. And in this case, there are now 3 samples, and they could coalesce or share a common ancestor in any of 3 orders. In this case, the sample from A is more closely related to the sample from B than either of them are to C. So there's 2 key things to notice about this model here. First is that the gene tree topologies don't need to match species tree topologies. And second, we see that the gene trees always have coalescence time or share common ancestors that are older than the species tree divergence times. So they can't coalesce until they're in the same population, which means coalescence times are older than the speciation times. So I just give an example of one tree. We could sequence lots of other loci, and we're going to get different gene trees. We might have different topologies, but we'll almost certainly have different coalescence times for different gene trees. And so this is the model I would like to use, but I'd like to incorporate tip dating. And so I want to have a case like this, where I can have species that are potentially extinct, and we can sample individuals from different species at different times. So the tips of the gene trees don't need to end at time present. So my goals here are to develop a multi-species coalescent model with tip dating, and then to implement this model in the program DPP, and then lastly to test this method's performance. So I'm not going to go into too much detail here, but there's a few key assumptions about this project. First, we're assuming the species tree is known. So this might be reasonable for something like elephants and mammoths. We think we know the relationships between elephants and mammoths. We're also going to assume that all of the samples have known ages. So what this means is for every mammoth sample, I can say, okay, I'm going to estimate this age with something like radiocarbon dating, and I'm going to assign it an age before I start phylogenetics. And then lastly, I'm going to assume that all samples are from tip populations. So if these dots are the samples, I can sample an individual in A, I can take a sample from B or C, but I'm never going to sample the common ancestor of A and B or the common ancestor of A, B, and C, because we're assuming we can assign the samples to a species a priori. So as part of this project, we can now calculate the probability of a given gene tree conditional on a species tree. So if I know this is a gene tree, I can calculate the probability density of this gene tree given the species tree. I'm not going to tell you how to do it, but this is some of the new stuff that's gone into this project. So some key questions I want to ask in this project are how well can parameters of the model be estimated, especially mutation rate and speciation time, and how does treating ancient samples as contemporary impact our inference results? And I'm asking this because this is something people do empirically. So up until now, you basically had to choose if you wanted to do the multi-species coalescent or you wanted to do tip dating. And so this led to people just ignoring the tip dates. So this is why we're asking this question. So the first thing I did was a simulation with an extinct species tree. So I have a root age here that's 10 million years old. Then I'm going to say that species A and C are extinct. I'm going to say they went extinct 5,000 years ago, and I'm going to draw their sample times between 5,000 years and 50,000 years ago. We're using 50,000 years because that's about as old as radiocarbon dating will work, and it's also just easier to get younger DNA sequences. So for every set of conditions, I'm going to simulate 20 data sets and do inference on those 20 data sets, and I'm going to be presenting averages across those 20 data sets. So first we looked at divergence time in expected number of mutations. So on the x-axis here, we have the number of loci. On the y-axis, we have our divergence time. And then each point is the mean of the posterior distribution averaged across those 20 replicates. And then the 95%, or the error bars are the 95% credible intervals, again, averaged across those 20 replicates. And what we can see is as we increase the number of loci, we get really pretty good estimates of our divergence times and expected number of mutations, and they appear to be centered at the true values. So the dashed lines are the true values. So this seems to be doing well and expected number of substitutions or mutations. So next we look at the same plot, but now we're going to plot it in years instead of mutations. And so the general trend is the same, whereas we increase the number of loci, we get better estimates, though our credible intervals may be a little bit bigger. So next I wanted to look at our inferences on mutation rate. So on the x-axis here, we have number of loci again. On the y-axis, we have mutation rate. And what we notice is that for small numbers of loci, for 10 loci, we have really poor estimates of mutation rate. This is basically the prior that we're getting back, which I'm not showing. But as we increase the number of loci, we're getting much better estimates of mutation rate that are much smaller than the prior. And I want to point out here, we are not using fossils in this analysis. So the only information we're getting on rate is coming from these ages of the samples. There's no other information. I also wanted to look at what could impact our inferences of mutation rate. So in the examples I was just showing, our samples were at most 50,000 years old. I also looked if they were younger, only 10,000 years old. I looked at if we had 3 or 6 samples in each extinct species, and whether we had 1 or 2 extinct species. And the trend we can see is we do much better if we have older ages of our samples. And we also do better with more samples and more extinct species. And this makes sense because all of these give us more dates or older dates. So I also wanted to do a simulation with a much more recent population divergence. And so in this case, the root age is only 20,000 years old. And for each population, we're going to draw samples between time present and the age of the parent node. So we only have ancient samples. And so for example, for A and B, the samples are going to be drawn between time present and 5,000 years ago. Again, we're going to do 20 replicate simulations and present averages across those replicates. So on the x-axis here, we have the number of loci. On the y-axis, we have the node ages. And we can see that when we don't have very many loci, we get pretty poor estimates and they're a little bit biased. But as we increase the number of loci, we get pretty good estimates of the 2 younger node ages in the tree, and they appear to be centered at the true value. So as we add more data, we get better estimates. Seems like the method is working well. And then lastly, I asked, what happens if we treat the samples as contemporary? We said, in 5,000 years, not that old, what if we ignored them? And here the story is different. Again, we have not very much information when we have not very many loci, but as we increase the number of loci, we get smaller credible intervals, and they no longer include the true value. We're becoming increasingly certain that our ages are too low. And so this is a cautionary tale that even if you have young sample ages, if you have enough data, you can start to see a bias in your inferences of divergence time. So lastly, I did some empirical analysis with woolly mammoths and elephants. So I have 4 species here. I have a woolly mammoth, which is sister to the Asian elephant, and then these 2 African elephant species. So I have about 70 mitochondrial genomes for mammoths and about 25 genomes for the elephants. And so there's a few things we notice with these results. First, this divergence time between the 2 African elephant species is really, really young. It's about 25,000 years old. And we think this is due to a recent history of hybridization between these species. They live in the same space. They weren't recognized as independent species until fairly recently. And it's been supported by outside work. So we're not too surprised about that. The other thing we notice is that these other 2 node ages are about 3 or 4 times younger than previous empirical analyses. And we think this is due to DNA degradation. So DNA, as it degrades, cytosine is systematically deaminated to become uracil, which is sequenced as thymine. So this is a transition. And if we look at our DNA substitution model parameters, we see the transition transversion rate is over an order of magnitude higher than typical empirical data sets. And this is a value that doesn't change that much for something like mammals. And so this is a really strong signal. And then if we look at our mutation rate estimates, they're also higher than we expect. So basically we think this DNA degradation led to higher estimates of mutation rate and then lower estimates of divergence times. And so we think this is a product of the DNA quality and perhaps suggests that more work should be done on DNA degradation. Lastly, I looked at a nuclear data set with the same four species plus an American mastodon. And these results, so I only have one individual for each species. We have about 350 loci, but they're only about 100 base pairs long on average, which is incredibly short for phylogenetics. And generally these results match previous results. We just have really large credible intervals. And we think this is probably because there's just not that much information in this data set, and perhaps we would do better with a larger data set. But there's just not that much nuclear DNA publicly available for mammoths at this time. So in conclusion, we have a method that accurately estimates divergence times and mutation rates when we have a correct model. We also see that increasing the number of loci greatly improves our inferences, and that treating samples as contemporary can bias our divergence time estimation. In the future, we'd like to be able to improve our method convergence with more loci. So as I mentioned, this is an MCMC program, which is, this basically means that because of some technical details about MCMCs, our method works much better with smaller data sets, and we would like it to work better with large data sets because we have more information in them. So this is something we hope to improve in the future. We'd also like to incorporate the estimation of migration into this program. So BPP already allows for the estimation of continuous time migration rates between species under an isolation migration model. And we'd like to be able to incorporate tip dating into this model. So right now you can do tip dating or you can do the IM model. We'd also ideally like to be able to infer the species tree, which again is something we can do in BPP, but not with tip dating at the moment. So with that, I'd like to thank various funding sources, NSF, grad studies, and NIH. And then thank all of my committee members. Of course, I'll thank Bruce. And unlike a lot of top bio seminars, I just have one page of pictures. But I'd like to thank all my lab mates, all my family members, and of course my puppies, and all the other people here at Pop Bio, my cohort in the office. So thank you, and I'll take any questions.
Anna, your HIV analysis requires that that within loci, there's no recombination. Is that true?
Probably not, but it's an assumption people make all the time.
Did you check?
Well, with our simulated data, there wasn't. There are recombination finder tools, but typically what people do is they just use short chunks. So that's why like some of these sequences are very short, and there's not very much information because they just use short chunks and hope for the best. It's also pretty hard to, it can be hard to tell depending on the amount of diversity within an individual if there has been recombination. So we don't necessarily know the recombination finders work to start with.
Yeah, that was a really amazing talk, Anna. Could you clarify for your DNA simulator, you assumed that there was no, in the latent HIV, there was no like mutation. And I think earlier you said like there was like in latent cells, there's still mutation, but just lower. Is it like so small?
Well, I think empirically we don't really know. It's probably similar to the human somatic mutation rate or like mutation rate of, there is clonal expansion of some of these latent cells. So they could potentially be replicating, but the human mutation rate is just so much less than HIV that even if there are mutations, it's probably not going to affect our inference more than all the other things that could affect our inference. So we could change that assumption, but basically the mutation rate is low enough in comparison to HIV that we didn't think it was necessary. Like when you sequence clonal expansions, the sequences are often identical.
Are you aware of whether there are any sort of clinicians or doctors who have used this method or anything sort of like it with patients? And so what's the sort of feedback you've heard? If there is?
So there's only a few labs that generate this type of data. It's very expensive and very tedious. And I think the people who use it are usually like HIV clinician researcher type people in academic settings. So I think people are using it in like a help patient sense, but more as a basic science, understand this reservoir sense, and I have not talked to any of them. Well, there's only a few labs. We're pretty sure they reviewed our paper. So yeah, there's probably like, so the methods we use, I think one of them was actually developed for a different purpose, but could be applied to this data. The other two were from the same lab. And so, yeah, I don't know why we haven't talked to them. We assume they read our paper and they've cited us, so.
Right.
I was curious, like, could you say a bit more about what types of sequencing data sets are collected? And I guess I'm curious about these latent cells and like, I'm guessing you're sampling these different types of sequences like virions and latent cells with very different like efficiency, and like does
Yeah,
something about the cellular biology of it impact any of the inferences that you make here?
So the virus sequencing is pretty easy. You can only sequence the latent cells when you are being treated effectively with your antiretroviral drugs. And basically, it's very inefficient to sequence them. And so you have to, it's called a quantitative viral outgrowth assay. You try to stimulate these latent cells to get them to spit out viral sequence. And you often have to do it multiple times. And people know it's really efficient and they know the more times you do it, you get more sequence, but I think we don't know that much about why. For latent cells reactivating, people think it might have something to do with where in the genome they're integrated. They don't integrate randomly, but some places seem to be better for keeping them latent versus not. So presumably there is some biology going on, but it's pretty poorly understood. Also, like out of your these are typically memory T cell, and it's like 1 out of 1 million will be latently infected. It's very small. So to sequence them, first you need to find some that are latently infected, stimulate them. You have to do everything at limiting dilution so you don't get 2 sequences in the same like sequencing pool. So I don't really know. I don't think anyone is that sure.
Right. Does that affect like, is it just affecting the amount of data that you get, or does it have any bearing on, yes, like the actual inferences that you draw, like what proportion of these different sequences you actually capture?
I think it would probably be mostly the amount of data, unless there was something about the actual sequence that made it more or less likely to be able to get the data, which I haven't seen suggested. What I have seen suggested is where it's located in the genome, which people think is not totally random, but like if it didn't vary over time, if the proportions of where you integrated at different time points were the same, I don't see why it would matter. But if like early infection, you had like a different trauma later, then it might matter.
Hey, yeah, that was super great. This was a question about a point that you purposely skipped over, so feel free to tell me to not, you can say no to this, but I guess I was curious about, you mentioned like you have this SDE model with the transitions between, for the HIV, right, the stochastic differential equation, and from that you got this like topological structure of a tree, right? So I guess I was just curious, like how does that work? Is it a SDE of like the?
So we're doing, so what we do is we simulate waiting time. So we calculate the rates of events. We simulate a waiting time to an event, and we decide what event it is. And then we basically are keeping track of the entire population of cells. And so we assume that all cells, like if there's, for example, uninfected cell becomes infected, any of the viruses are equally likely to lead to a new infection. So we just draw one of the viruses to the branch, and then that will be the one that you draw the new lineage from, if that answers your
Yes, that makes sense.
and for HIV, we don't necessarily think this is totally true. There's a ton of selection in HIV, but this was our first pass at it.
Cool, thank you.
Well, thank you so much, everyone. Let's, well, stay